Hello, I'm Matt, and I'm an MSc student in the Decoding Lab at the University of Calgary. I will be walking you through my poster presented here at the Great Lakes Bioinformatics Conference. Approximately 2/3 of missense mutations are believed to be deleterious in humans. While most pathogenic mutations are believed to be deleterious, it is not known if the reverse is true. Variants not considered pathogenic are typically considered benign by clinical geneticists. However, usually only in the context of a specific disease, not necessarily in general. A common pursuit in variant classification is the training of machine learning methods to classify a variant as pathogenic or benign. Ideally, classifiers would be trained and evaluated on generally benign variants as negative cases. A potential source for these variants would be those that are evolutionarily neutral. Typically, neutral variants are selected as having an allele frequency greater than 1 to 5% in population databases. However, this assumption is rarely substantiated. The purpose for this project was to simulate the expected distribution of standing genetic variation in modern human populations and assess the misclassification risk when designating a variant as neutral when using various allele frequency thresholds. We quantify this risk by neutral odds ratio, which is simply the odds that a variant at or above a threshold is neutral rather than deleterious. Moving into our methods, I will first address several of the models we use in our simulations. Firstly, we adapted the distribution of fitness effects derived from ESP by Kim et al. For simulating human demography, we adapted two similar models. Firstly, the Gravel et al. Model derived from Thousands Genome Project, and the Tennyson et al. Model inferred from ESP. Lastly, we derived our evolutionary parameters from the Thousands Genome Project. Our first approach was using SLIM to simulate a 1 megabase locus across 5,000 replicates for 4 different recombination rates incorporating the previously discussed models. Secondly, we then calculate the expected allele frequency distribution in modern humans with the Wright-Fisher exact solver developed by Dr. Ivan Krukov in our lab. Here we address the effect of penetrance by introducing it as a random effect in the Wright-Fisher model. Additionally, we consider both additive and recessive fitness models while simulating population changes through a discretized model of human demography. Now for our results. Firstly, we found that reliable detection of neutral variants is really only achievable in regions of the genome with high recombination rates. Conventionally, false negative rates of around 5% are desirable, which corresponds to a 20x odds ratio. In an African population, a 20x neutral odds ratio can only be achieved at an allele frequency of around 60% in regions of average recombination, and in 30% for regions that exhibit the top 95th percentile rates of recombination. The result is even more stark when simulating the European demographic history, where strongly deleterious variants are more highly enriched in the modern population than what we observed in Africans. Here, a 20x odds ratio can only be achieved in regions with the highest recombination rates and at allele frequencies around 65%. Even 10x neutral odds ratios are unachievable for low and median recombination rates in both populations. I have summarized those findings here, where we can see that at a conventional allele frequency thresholds, we are only achieving at most 6x odds ratio, which corresponds to a false negative rate of 14%. We also illustrate the effect of constructing a dataset of neutral variants using these various thresholds, where striving for a 20x neutral odds ratio means a large reduction in the number of variants available, but are significantly more depleted of deleterious variants misclassified as neutral. The last finding I will be discussing today is the impact dominance and incomplete penetrance have on a deleterious variant's ability to persist in the population. Perhaps unsurprisingly, deleterious variants under a recessive model are able to achieve remarkably high allele frequencies in the population and is further compounded by incomplete penetrance. The same is true for variants under an additive model. However, the more strongly deleterious variants at least achieve 20x neutral odds ratios at much more reasonable allele frequency thresholds. To summarize, our simulations suggest relatively higher allele frequency thresholds are needed to confidently classify a variant as neutral. The most important consideration is how confident one must be for a given application. By reasonable standards, certainty can really only be achieved for variants in regions of the genome with high recombination rates at very high allele frequency thresholds. And lastly, recessive fitness and incomplete penetrance further compound these effects and allow deleterious variation to persist in the population at high allele frequencies. That concludes my presentation. I would like to acknowledge our funding sources for this project, and I would like to thank you for listening. I'd be happy to answer any questions you may have at my poster station.